lunch though. Any doubts? No doubts? Are you sure? I'm going to ask you questions. Okay. All right. So, we're supposed to delve into chromatin organization today, but we'll at least begin. We might finish tomorrow. There's a lot of stuff in this space. This field has only evolved in last. Acha. 2009. A key author in the first human genome that was sequenced in 2001. If you make discoveries that are amazing, changing biomedical science, then you get to become scientific advisor to Obama, right? Cool? So, Eric Clinton's a great guy, an amazing scientist, an amazing administrator who made Broad Institute from nothing to what it is today. Maybe I should look at the Broad Institute.
Excuse me.
नहीं हो रहा है? First, human genome sequencing has a lot of value, right? For what we do today in terms of biomedical science. He was the one who headed the Cancer Genome Atlas project, which made big discoveries into cancer genome changes. What is wrong in cancers? That he discovered a lot of that, and he led that whole discovery. Billions of dollars, millions, and not billions of dollars went into sequencing cancer genomes after the discovery of that. And then we have discovered a lot of those proteins that I tell you about. 20% of cancers have PTEN mutation, 20% of cancers have KMT2D mutations, I told you, right? All of that came from the Cancer Genome Atlas project. So we now know how to treat some of these patients, which is brilliant, right? I told you over large number of cancers have MTAP mutation in which PRMT5 inhibitors are used, etc., etc., etc. Lots of such discoveries where we can now know exactly what patients might have, what kind of genetic alteration that we can give to them. So the point being, discoveries, very impactful discoveries can be made by being good thinking, by the sequencing that we talked about, right? The SBS, that discovery was made in Cambridge, so they utilized this and sequenced. There's an institute in UK called the Cancer Research UK that is funded by Wellcome Trust. It was funded by Wellcome Trust. Wellcome Trust is a big funding organization that funds a lot of research, including in India, actually ties up with the Department of Biotechnology. So these are very important things as some of you, if you think of becoming policy makers later on for Indian government or other governments or, help out in that, you need to think about the future as to how we shape the next biomedical science, next discoveries, okay? That's the critical piece, all right? Okay, so he made a very important discovery in 2009 because he saw the human genome. He said, oh, how is the human genome folded? Let's figure this out. So the first author of this first paper is called Erez Lieberman. He's a professor in Baylor College of Medicine, which is next door to where I work. And I collaborated with Erez, I've collaborated with Yob, and we've looked at the structures. We'll talk about these things as we go along. But today, the important thing is, the question is, how do chromosomes look, right? We talked about how is that whole 2 meter of DNA packaged into chromatin, which we'll brush our understanding how do chromosomes look like. And again, as I said earlier, chromosomes are only in metaphase, right? They're not there everywhere. So what does this slide tell you? This slide is telling you the states of cell division, right? So interphase, on the first day I emphasized again and again and again, interphasic chromatin structure is different than the chromatin structure in the rest of the division states. Most of our cells are in interphase stage. Only very, very few of them are in any of these stages. So it's more important for us to define what happens in interphase first. And then there was a landmark paper where Job Decker and others described how these 3D chromatin structures change as the cell goes through all of these different cycles. And it turns out that the cell opens up the whole chromatin structure and reorganizes it again, which makes sense, right? I mean, it's going into the chromosome comes out, then everything. So the whole 3D chromatin structure goes through a process as we go through the 3D, we go through the replication process, okay? Okay, so an interphase, there is that. He's showing you things on the top, right? So 10 nanometer fiber becomes a 30 nanometer fiber, a 300 nanometer fiber becomes a chromosome. And then we need to understand the levels of chromatin organization. We know this. Everybody knows this. What is this? Okay, what is a nucleosome? DNA wrapped around histone together. It's called nucleosome. Otherwise, the octamer is called histone octamer. Histones look like which have 3 folds, 3 alpha folds. That's what it's showing here to you, right? Okay. So a nucleosome is a building block of chromatin. You know this. This is how histones look. We're just brushing our knowledge again because you guys get confused. And I just took the slides. So histones are highly basic proteins, abundant in lysine and arginine residues. You guys know this. You've seen this slide earlier. We've talked all about histones. Histones are there. These are the histones, H3, H4, H2A, H2B. Look at the slide again. Make sure you know what it is. They look very similar, right? H3 and H4 make a dimer, and H2A and H2B make a dimer. Interesting. We'll talk tomorrow about histone chaperones. The histone chaperones that uniquely take H2A, H2B away or H3, H4 away. We'll talk about histone variants at some point. Histone variants are the ones that are not regular histones. Okay. I love histones. I love nucleosome. I love chromatin. Right? It's beautiful stuff. So these go together. H3, H4 dimer, H2A, H2B dimer. That makes the nucleosome. That's a fundamental unit of chromatin architecture. Must appreciate histone octamers. Histones. H3, H4 tetramer, H2A, H2B tetramer go together, makes an octamer. I sometimes also keep my password as histones. Easy to remember. Love of your life. Of course, after my wife, right? My daughter was asking me the other day, can you send me, where is the YouTube? I want to look for your lectures. And I was like, I'm not going to tell you because I'm telling you all of them about your things that you do. Anyways, okay, around 200 base pair, you know all of this. We're not going to get into this again. 1.67 left-handed superhelical turns around histone octamers. Beautiful positive negative charges. The bead on the string, we know this. Okay, good. 10 nanometer fiber, 30 nanometer fiber, right? Okay, one of the things that people thought about is how is this arranged? A 10 nanometer fiber can go into different ways to make the 30 nanometer fiber. We didn't talk about this. I took this out intentionally because I didn't want you guys to be confused. You see, I keep giving you the same information again and again, right? Because your brain will process it, you'll remember it with your long-term memory. There's a solenoid model you can read, and then there is a zigzag model, right? In a solenoid model, the interactions happen between consecutive nucleosomes, and in zigzag model, interactions happen between alternate nucleosomes. Is this really important? Is it? Why?
Because for packaging of DNA, it has to undergo all these folds, otherwise it would remain as a tangled thread in the.
Wonderful, right? It's very important how these things go together. We don't know how to drug it. One day, my division head says, Kunal, amazing. How do you drug these chromatin states? I was like, okay, good. Let me think about it, right? I have not entirely found an answer, but that was a good thing. One sentence from the guy who treats a lot of melanoma patients. He's the current CEO of a institute called Moffitt Cancer Center in Florida. His name is Patrick Hu. He used to meet with me once a month when I started my career, and when I started my independent job, he's like, what an amazing amount of work you do, right? And he got half of it, and he didn't get half of it, just like you got half of the chromatin states and not the other half, right? Maybe you got more than him. But he was very fascinated with what I was doing, and he said, we must find a way we drug these nucleosomes. That's why the first slide I showed you guys was a dragon of nucleosomes, remember? Yeah, because I love that, right? How can I drug it? That's my problem. That's what keeps me up in the night, right? So you need to find what keeps you up in the night. Beyond the girl you love, beyond the boy you love, right? Okay. I make funny jokes. Is that good? Right? It's very important. It's very important that we love somebody. We love our families, right? Of course, your parents, your wives, your husbands, your girlfriends, and the boyfriends, and the kids you have, and the grandkids you will have. Now, where am I going with this? Zigzag and solenoid models. Let's focus here, right? This is important, right? Because this creates a structure that we could think of for tomorrow as to how to drug Mr. 30 nanometer fiber. I don't know. I still don't know the answer. Okay? Do I need to have an answer? Possibly. But first, I need to understand how does a 3D chromatin structure go wrong in cancer, because that's what I study. You can look for how does the 3D chromatin structure go wrong in diabetes. Maybe it does. Or 3D chromatin structure goes wrong in hemophilia. You guys know about hemophilia? Yesterday, I met a friend of mine, and he says his friend is scared. A 10-year-old kid has hemophilia, and they have to give him 1 lakh rupees injection every month, and they cannot afford it. The guy is dying. How do we figure that out? Right? What is hemophilia? Yeah, your clotting does not stop, right? Does not happen. I'm sorry. That's right. See, I can learn from you too. Clotting does not happen, so you have to give them all those factors, coagulating clotting factors. So that's the injection he's talking about, right? Can I do something about that? Can I? I'm just throwing it out there just because I heard it yesterday night, right? But there are lots of diseases where there are implications of these, and we'll talk about diseases after some time, maybe tomorrow. The other professors who will come later, they'll talk more about diseases than me. My job's to tell you basics of epigenomics, but I do bring in a lot of diseases, and I don't want to take the thunder away from my fellows who are covering up for me afterwards, right? Then we have 300 nanometer fiber, right? How does this structure form? I don't think people know. Okay. Yob and others have tried to figure out, and Altra is still figuring out. There are models, they've figured out some things. And I think one of the things, at least I understand, yeah, here, let's go there, right? So we covered the right side, the left side, the 300 nanometer fiber, and then the 700 nanometer fiber. I don't think people really understand how these things happen because we don't have technologies. These require a lot of sequencing and a lot of money, okay? I do some of these experiments in my group. I actually do a lot of it, and it takes so much money because you need to have lots and lots and lots of sequencing, because in histone modifications only happens in 1% of the nucleosome. So I don't need more than 30 million reads. But if for this, I need 2 billion reads. And to do 2 billion reads in a sequencer, I need to spend a lot of money out, okay? I'll come to high C in a second if we have time today, or I'll come to it tomorrow, you'll see why. But in a chromatin structure, it's genome-wide, right? So I need to have the whole genome sequenced again and again and again and again to be able to cover for the whole genome to understand these architectures. Very few labs have that kind of money to do it, okay? And NIH does not want to fund that. Mr. Trump does not want to fund it unless, and India doesn't have enough money to fund it. But China does. Okay. So what factors determine these, right? I mean, I just showed you what is just simplistically overview. I'll go deeper into it tomorrow. I just don't have the slides right now. Okay. But what factors determine these chromatin condensation that we just observed, right? There are factors that actually put these, you saw the rings now? Rings, these rings are called condensins and cohesins. Have you heard of cohesins and condensins? Okay, cohesin. Cohesin is a tripartite structure of 3 proteins, SMC1A, SMC3, and RAD21. Okay? There's like rings. They open like this, they close like this. They open like this, they close like this. Beautiful again, nature, right? Nature is beautiful. Thread, you put the clasps around your whatever you want to put together, right? Like your curtains, put them together through the clasps, right? That's what it's doing. It's trying to put things together in place by putting a ring-like structure. There's a beautiful, very, very amazing scientist called Kim Nesmith in Austria, who's done a lot of work on cohesin and condensins. Should look at some of his papers. Fantastic scientist. I used to really admire his work when I was an undergraduate. Okay, just like you guys, some of you guys should look up his work. Condensins, cohesin condensins. They require ATP to open and close. So energy to open and close, right? Cool? Condensins actually are mostly just important during the cell cycle. Cohesins, on the other hand, are also very important to keep the interface nuclei structure together. Okay, so we find a lot of evidence, not me, others, obviously, find a lot of evidence that cohesin is very important in maintaining the 3D looping that we talked about earlier. Remember the 3D loops I told you was CTCF? So CTCF and cohesin work together very closely to keep these loops together. I don't think it's here. I will send you the slides. I'll show that tomorrow. I work on cohesin a lot, actually, because we find that cohesins maintain or drive or specific forms of cohesin, a specific acetylated form of cohesin, actually drive melanoma metastases. Remember in the slide I was showing you, I also studied metastasis. Metastasis is how patients die. Patients die because the disease has metastasized to different organs, to our essential organs. You don't die if your melanoma is here, it doesn't matter. Melanoma spreads to the lung and the liver and the brain. That's when people die. So the key thing is to solve metastasis, right? And we find that when you have an unacetylated form of cohesin that cannot close this ring well, actually promotes metastasis. How cool is that? Amazing, I love it, right? And that is done by an enzyme called HDAC8, histone deacetylase 8. It doesn't do histone deacetylation, it actually deacetylates cohesin. Okay? So a lot of these enzymes that I'm talking to you about who does histone function actually have non-histone functions as well. That's what I wanted to get you to. Okay? They actually can modify other non-histone proteins also. It's another field of epigenetics completely. Okay? A lot of those proteins have activities on non-histone proteins. We are just talking, we're going to stick to epigenetics, so we're going to stick to histones, but it's a growing field. It has, there's a lot of beautiful stuff. P53, I'm sure you've heard of P53. P53 gets modified by a lot of these. It's a transcription factor. The most mutated gene in cancer world is a transcription factor, P53, absolutely. almost all cancers have an influence of p53. It's a transcription factor. There is no better evidence than that epigenetics is critical for disease setting, right? I hope that you appreciate that fact. In fact, most of the drugs that are in clinical world right now are transcription factor targets. Have you heard of anything called androgen receptor? Prostate cancer. Estrogen receptor, breast cancer. Progesterone receptor, or whatever, right? The next PRs. Nuclear receptor, transcription factor. All of these are transcription factors. They are nuclear transcription factors. Okay? HER2 is not. But ER, PR are. Okay? Nuclear transcription. We have drugs against these. So all the patients who breast cancer patients who come to clinic, ER positive, PR positive, they're given things that make these transcription factors not work. Right? You get it? That's actually a lot of those are. So a large number of drugs that we give to patients today are actually working on transcription factors. We knew this for a long time. So anyways, the transcription factors are critical for cancer. I mean, I'll keep talking about cancer because I don't know enough about other diseases, unfortunately. But okay. So there is this enzyme called APC, anaphase promoting complex, that inhibits securin. Securin, along with separase, I think works on these things and breaks these rings during the metaphase. And then you can, in anaphase, there is no more that, and then that helps you separate them, right? Cool? These rings are critical to maintain the chromosome architecture. You've seen this. Electron microscope. Chromosome looks like a chromosome, otherwise it looks like a chromatin. We have euchromatin, we have heterochromatin. We've talked enough about it. We're just going to talk again about it, unfortunately. The entire 10 nanometer structure is not transcriptionally active, we know that. Transcriptionally inactive, transcriptionally active, right? What determines the differential response? Histone modifications, histone variants, we'll talk about that tomorrow, okay? Nucleosome remodelers, we'll talk about them tomorrow. Beautiful enzymes. Then DNA methylation, right? We talked about it. So heterochromatin, how euchromatin is different from heterochromatin, densely packed, loosely packed, transcriptionally active, transcriptionally silent. I think we know all of this. How is euchromatin different from heterochromatin at a molecular level? Different histone modifications. Now we've got this. Different histone variants. We'll got that tomorrow. And then differences in DNA methylation, right? Cool? Summary, good summary of all you've learned so far. No confusions. Histone modifications. Why am I talking about histone modifications? Because he just wants us to remind us that acetylations, methylations of specific ones, and phosphorylations are found in the euchromatin again, right? And other modifications like K9 dimethyl, K9 trimethyl, H4K20, and H3K27 methylation are found in heterochromatin. According to him, some of the K27 is also in the euchromatin, we know that. Histone variants. Oh, he does have a slide here too. H2AZ is a histone variant of H2A. Easy peasy. Does have few amino acids here and there different. H2AZ is only found on genes that are to be transcriptionally active, become transcriptionally active. H2A point Z. So it's in euchromatin, just like H3K4 trimethyl. Remember, I told you H3K4 trimethyl is only there on transcription on genes that are supposed to become active, that can become active. So is H2AZ. Okay, so H2A is replaced by H2AZ by some specific proteins called histone chaperones. H3.3, that's another one. Histone variants. H3.3 can be there, deposited on replication forks. It actually tracks well with where replication happens. Okay, so cells have made these beautiful things in their mind. We're gonna put specific marking on histones itself beyond just the modifications to be able to recognize where we're gonna do what. Okay. रिप्लिकेशन में क्यों होता है? 3.3 ट्रांसक्रिप्शन के टाइम में नहीं होता है. I think replication के टाइम में होता है, if I remember correctly. Okay, let me look it up and I'll get back to you. I don't work on histone variants much, so I actually always don't have enough knowledge. Okay, but let me look it up today. So in H1s I talked about the first day. They're only found in certain places, and they're actually found in specific places where it's usually found in heterochromatin because it allows the compaction of the chromatin. Remember, we talked about chromatin compaction, we talked about PRC1. So PRC1 actually associates with this, and it associates with SUV complexes, and those actually pull up histone H1. Histone H1 is at the entry-exit site where DNA enters and exits. That's where H1 is bound, right there. Okay, cool. H1 is much more enriched in the heterochromatin than euchromatin. That makes sense. People do not have sufficient understanding where and when H1 works. Still a little bit of an enigma. It's a different kind of histone altogether. Okay, so whatever he's drawing is, there is not enough. Suppose there is some loose evidence for it, but I think there is still debate in the field about how, where, and all histone H1 works. But for your knowledge, you can just say histone H1 is more in the heterochromatin than euchromatin. Okay, and to be honest, I still need to look at what's latest in histone H1 world. But some specific chromatin remodeling machines that we'll talk about tomorrow actually associate with histone H1. So they have to open that gate because now it's blocking. It's like a lock on a gate. Okay, so you've got that, it's locking you in the position. That H1 interactions then allow the two histones to come together and have chromatin compaction or something like that. So I need to go back and look it up, but you guys should know that H1 is in heterochromatin, it acts as a lock, and gate lock sometimes, if you will, right, on heterochromatin. Heterochromatids are hypermethylated. You know that. We talked about DNA methylation, mostly hypermethylated, DNA methylation, hypomethylated euchromatin. That's not entirely true, as we know. Some of the methylation is in the gene body, is in the euchromatin, right? Okay, good. All right, access to promoter, non-access to promoter. When there is, histones are nucleosomes are next to each other. Of course, there is no access. Whereas in the case of euchromatin, there is access, so it cannot bind. We talked about this earlier. RNA polymerase can go through. Chromatin accessibility is important for transcription. I don't think that's news to you guys. Is not? Good. Regulators of chromatin accessibility are called chromatin remodelers. We will again talk more about it tomorrow. I'm just going to introduce it right now here. Chromatin remodelers are large complexes of many, many proteins. In fact, my PhD advisor discovered the first chromatin remodeler. How cool is that in Roger Kronberg's lab? So he worked for first 10 years on just characterizing a chromatin remodeler called RISC in yeast. And I did some project with him as well, and his whole lab worked on yeast. But this is chromatin remodeling complexes are amazing because they actually can utilize the energy of ATP through their ATPase domain to move the nucleosome on the DNA. Moving that huge piece of nucleosome that you saw on a DNA is a lot of work, requires energy, right? So we didn't know how this works. What happens? There are many ways it can happen. Okay, nucleosome remodeling complexes can bind, and new sites become accessible. What did it do in this picture? What is it trying to show you? What are these? Huh? He's saying nucleosome came in and it threw the histomer out of this. It's called eviction, right? What does he call them? Stone eviction. That's right. I was right. Stone eviction. So he removes one of the mechanisms is nucleosome remodeler. Some nucleosome remodelers work is by histone eviction. I will cover this tomorrow again, okay? But essentially nucleosome remodelers can work by multiple mechanisms. One of them is throwing the nucleosome out of the DNA and saying, boom, I need this site open, guys. So that's nucleosome remodeler, one. And then two, it can do a histone replacement. Okay, so because it carries histone chaperones. Chaperones do what? They exchange the histone in the nucleosome. Okay. And then it can do nucleosome sliding. It can slide the nucleosome from here to there. Make sense? And then he is not talking about a specific mechanism that is called histone nucleosome translocation. What does translocation mean? Similar to sliding, but there what happens is, this is the nucleosome. Let's say the DNA is right around this. It opens, the nucleosome comes out, it goes on, tracks along, opens the nucleosome. That's, you got this? Very simple, right? That's called nucleosome translocation. It's not same as sliding, but that's a translocation. So it tracks specific number of bases. It opens exactly the same amount it wants to open. Okay? Okay? So that's a chromatin remodeler. Cool? Okay, I'm supposed to teach you ATAC-seq tomorrow. Just like ChIP-seq, there's something called ATAC-seq. ATAC-seq is something that looks at chromatin accessibility, open chromatin. Where is open chromatin in the genome? And that allows us to identify transcription factor binding sites. So whenever it's an open, that means something can bind there, right? And there might be a sequence enriched there in that place. And then if you look at the motif analysis, you can say, okay, there is motif enrichment here of X transcription factor. That means that, let's say there are open sites that are in one condition versus not. Let's say in non-tumor versus tumor, right? There are ATAC open sites. You can say, okay, maybe this transcription factor worked compared to the others. Okay? We'll talk about it tomorrow again. I think I need to take you guys through ATAC-seq step by step. I'll do that tomorrow. ATAC-seq is a very, very, very popular method now because it's very, very, very easy to do. And it was a very, very smart use of transposase. Have you heard of transposases? No. Transposase are things that transpose things. They're viral elements. Viral elements, I told you earlier, right? Go from hop on from here to here. Internally, viral elements code for transposes. So the transposes, when they code for, that can go bind to itself and or its DNA, and it moves that to the next place. That's called transposition. Transposition. That's how viruses work. Viruses hop around your genomes through this mechanism of using transposition. So there's a guy called William Greenleaf in Stanford. He identified a method called ATAC-seq. Devised a method called ATAC-seq by using a specific transposase that came from a virus TN5. It's called TN5 transposase. We'll talk about it tomorrow again. Very simple technique. You put a transposase, you put a specific barcode that you want to put in a sequencing reaction, right? He just says, okay, let's take the barcode that I put in sequencing reaction, combine it with the TN5, put it in a reaction, and the reaction will automatically taste. It's going to just put those barcodes anywhere it finds an open chromatin, because transposase cannot jump to a place where there is a nucleosome. Of course, it cannot, because there is steric hindrance, right? What does a steric hindrance mean? You see that steric hindrance means in space they cannot go there because there is already nucleosome bound to the DNA, and the DNA is not open, just like where transcription factors cannot bind, TN5 cannot bind, of course, right? It doesn't have access to it, right? But TN5 will go wherever it finds an open chromatin. It's just going to go deposit the barcodes there. It naturally deposits that.